بسم الله الرحمن الرحيم. Our video today is about thymic stromal lymphopoietin in diseases other than allergy. And again, I'm reminding you to subscribe to the channel and activating the alarming bell if you are interested in getting any new video. And this will be the third video about the role of thymic stromal lymphopoietin. And we'll start by its role in infections. And again, I already summarized the information that came from this nice paper, Role of Thymic Stromal Lymphopoietin in Allergy and Beyond, in Natural Review of Immunology, published 2023. The expression of thymic stromal lymphopoietin is high at body barriers, including the skin. Thymic stromal lymphopoietin acts directly on neutrophils to increase their killing capacity of MRSA. Also, thymic stromal lymphopoietin has been found to increase the efficacy of killing Streptococcus pyogenes, which is a significant contributor to skin infections. This is achieved by thymic stromal lymphopoietin directly interacting with the complement C5 system, which regulates the production of reactive oxygen species by neutrophils. As a result, thymic stromal lymphopoietin promotes the killing of MRSA in a manner that is dependent on both neutrophils and complement. Thymic stromal lymphopoietin also has an impact on the performance of immune cells as well as tissue inflammation and the ability of the host to develop protective immunity against helminth infection. Administering monoclonal antibodies to thymic stromal lymphopoietin in mice demonstrated that thymic stromal lymphopoietin is an important factor in the creation of protective T helper 2 cell responses following helmintic infection. And severe intestinal inflammation was observed due to the upregulation of interleukin 12 P40, interferon gamma, and interleukin 17A expression, which resulted from the lack of thymic stromal lymphopoietin signaling. Also, innate lymphoid cells type 2 play a crucial role in fighting against helmintic infection by producing T helper 2 cytokines. And there is evidence from recent research that the activity of innate lymphoid cells type 2 can be modulated by various neurotransmitters and neuropeptides such as catecholamines, nicotine, acetylcholine, neuromedin, vasoactive intestinal peptide, and calcitonin gene-related peptide. This finding already suggests a relationship between the nervous system and the innate immunity at the interface of barrier tissues. They found also that following infection with certain parasites or treatment with alarmins or cytokines such as interleukin 25, 33, and thymic stromal lymphopoietin, activated innate lymphoid cells exhibited a heightened expression of choline acetyltransferase, the enzyme responsible for the biosynthesis of acetylcholine. So, thymic stromal lymphopoietin has the ability to activate innate lymphoid cells type 2, leading to an increase in choline acetyltransferase production, which can serve as a mechanism to enhance the host's defense against helmintic infection. As regard the role of thymic stromal lymphopoietin in viral infections, while there has been significant research on involvement of thymic stromal lymphopoietin in T helper 2 type response, its role in CD8 positive T cell responses has not been extensively characterized. And we know that the main viral resistance is through CD8 positive T cells. However, in certain viral infections, like for example, coronavirus disease or COVID-19, which leads to severe acute respiratory syndrome, it is linked to the cytokine release syndrome, and this condition is marked by inflammation associated with T helper 1 and T helper 2 cells. And they found that in individuals with COVID-19, the production of thymic stromal lymphopoietin is triggered and elevated levels have been linked to more severe disease. And as a result, thymic stromal lymphopoietin appears to contribute to pathological processes and could be a promising target for therapies aimed at suppressing T helper 2 cell reactions in COVID-19 patients. So, apart from helminthic infections, thymic stromal lymphopoietin is involved in various infectious diseases, including viral and bacterial infections. And thymic stromal lymphopoietin induced by microbial infection might have a significant role in worsening allergic diseases such as asthma in such cases. As regards the role of thymic stromal lymphopoietin in cancer, here is a nice paper about thymic stromal lymphopoietin isoforms, inflammatory disorders, and cancer, which is published in Frontiers Immunology, July 2018. In the last 10 years, research has shed light on the roles of thymic stromal lymphopoietin in regulating the development and progression of various cancers, either solid tumors or leukemias. These studies have revealed that the effects of thymic stromal lymphopoietin on tumor growth can be either beneficial or harming depending on the specific tumor type and the surrounding microenvironment. Actually, thymic stromal lymphopoietin was found to exert diverse effects on both innate and adaptive immune cells, such as dendritic cells, innate lymphoid cells type 2, T and B cells, natural killer T cells, and Treg cells, eosinophils, neutrophils, basophils, mast cells, and macrophages. And as these cells are involved in tumor initiation, growth angiogenesis, and lymphangiogenesis, it is understandable that thymic stromal lymphopoietin can have direct or indirect impact on controlling various human and experimental cancers. Thymic stromal lymphopoietin has been linked to both promoting and inhibiting cancer growth in various types of malignancies, indicating that its impact in malignant diseases may vary depending on the specific context. Now I'm going to talk about thymic stromal lymphopoietin and fat metabolism. Here is a nice study that was published in Science 2021, July. Its title is Thymic stromal lymphopoietin induces adipose loss through sebum hypersecretion. Obesity carries a significant threat to public health, with more than 40% of adults worldwide being classified as overweight or obese. They are at heightened risk of developing chronic illness, including but not limited to type 2 diabetes, fatty liver disease, heart disease, various cancers with a risk of increase up to 7 times. Chronic low-grade inflammation resulting from obesity can contribute to the onset of insulin resistance, which can lead to type 2 diabetes. However, regulatory T cells or Treg can counteract this effect by suppressing inflammation caused by the obesity and maintaining insulin sensitivity, regardless of weight, and thereby preventing the development of type 2 diabetes. Research has indicated that the persistence of inflammation in adipose tissue leads to an increase in type 2 immune cells, such as innate lymphoid cells type 2 and eosinophils. And these cells play a role in boosting metabolic rate by encouraging a process in the adipose tissue, which is called beiging. Beiging means browning of white adipose tissue that involves the expression and activation of the brown fat specific genes in white adipocytes, and so increasing the thermogenic energy consumption because the brown fat has more thermogenic effect. So recent research suggests that the immune system may have the ability to control overall metabolism. T cells stimulated by thymic stromal lymphopoietin can trigger the loss of specific white adipose tissue, leading to the benefits such as obesity prevention, improved glucose metabolism, and reduce the risk of non-alcoholic steatohepatitis. In the study done in the mentioned paper, they found that changes in food intake, absorption, or energy expenditure did not lead to reduction in adipose tissue. Instead, the loss of adipose tissue was triggered by the excessive release of lipids through the skin via sebum. The steady-state regulation of sebum release and sebum-associated antimicrobial peptide expression was controlled by thymic stromal lymphopoietin and T cells. However, since exposure to thymic stromal lymphopoietin in the skin can lead to a reduction in size and lipid content of sebaceous glands by innate lymphoid cells, there is a possibility that innate lymphoid cells and T cells regulated both by thymic stromal lymphopoietin may have contrasting roles or opposite roles in the regulation of sebum secretion. In more recent studies, also it has been demonstrated that thymic stromal lymphopoietin has the ability to raise the number of systemic Treg, and as a result of this, thymic stromal lymphopoietin may have a counteractive effect on obesity and its related complications by expanding both type 2 immune cells and Treg. The basal metabolic index in humans was found to be associated with the concentration of thymic stromal lymphopoietin in the blood, and systemic energy balance is known to have a correlation with sebum production. When caloric intake is restricted, sebum production reduces significantly within 1 to 2 weeks. Also, alterations in the composition of the diet affect the composition of sebum. And while it's not possible that sebum release regulates the body weight as a homeostasis, it's possible to induce selective loss of white adipose tissue by increasing sebum release. This could involve diverting lipids towards the skin and away from the liver, which may also result in decreasing obesity-related non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. We have to notice also that the likelihood of developing asthma is higher in individuals who are obese due to the amplification of bronchial hyperreactivity, and this will result in poor asthma control. Also, patients with metabolic dysfunction who are obese tend to exhibit more severe asthma symptoms. So this can go back to the increase in the production of thymic stromal lymphopoietin and T helper 2 cytokines. Now I'm going to talk about the role of thymic stromal lymphopoietin in chronic inflammatory disease. Recent findings suggest that thymic stromal lymphopoietin may play a role in chronic inflammation and autoimmune disorders that go beyond T helper 2 cells, indicating its involvement in responses beyond the T helper 2 type. The first example is chronic obstructive pulmonary disease. Chronic obstructive lung pulmonary disease, or COPD, is commonly linked to helper 1 cells, macrophages, and neutrophils, while T helper 2 cells, eosinophils, and or mast cells are mainly associated with asthma. However, despite being primarily associated with T helper 1 cells, thymic stromal lymphopoietin messenger RNA and protein levels were found to be higher in bronchial epithelium of individuals with COPD compared to those without the condition. Agents that are recognized to aggravate COPD, such as respiratory viruses or cigarette smoking, and the pro-inflammatory cytokines that activate NF-kappa B are believed to stimulate the synthesis of thymic stromal lymphopoietin in COPD patients. This suggests that thymic stromal lymphopoietin may play a role in the development and or worsening of COPD. The second example is idiopathic pulmonary fibrosis. Idiopathic pulmonary fibrosis, this is a severe, progressive, and fatal condition that is distinguished by interstitial fibrosis of the lung, and its root cause is not yet known. However, thymic stromal lymphopoietin and thymic stromal lymphopoietin receptors are expressed at abnormally high levels in the lungs of individuals affected by idiopathic pulmonary fibrosis, and thymic stromal lymphopoietin has been found to be elevated in both bronchoalveolar lavage and serum samples collected from these patients. Also, the finding that antifibrotic treatment resulted in a decreased level of thymic stromal lymphopoietin in lungs of patients, while no such decrease was observed in individuals with advanced disease, suggests that thymic stromal lymphopoietin may be involved in pro-fibrotic type 2 immune responses in idiopathic pulmonary fibrosis. Rheumatoid arthritis is an autoimmune disorder marked by joint inflammation, which is chronic synovitis, predominantly linked to T helper 1 type and T helper 17 type inflammation. A recent study has shown that a specific variation in the thymic stromal lymphopoietin gene contribute to an elevated risk of rheumatoid arthritis susceptibility. These genetic variations are associated with increased thymic stromal lymphopoietin levels that correlate with rheumatoid arthritis susceptibility. In individuals with rheumatoid arthritis, levels of thymic stromal lymphopoietin and tumor necrosis factor in synovial fluid and plasma are higher compared to control patients, and that synovial fibroblasts can produce thymic stromal lymphopoietin in response to tumor necrosis factor not only in individuals with rheumatoid arthritis but also in control patients, indicating that tumor necrosis factor induced thymic stromal lymphopoietin production by synovial fibroblasts is not exclusive to rheumatoid arthritis and is heightened by tumor necrosis factor. Also, neutralizing anti-thymic stromal lymphopoietin antibodies have been shown to improve tumor necrosis factor mediated experimental arthritis injury, implying that a possible involvement of thymic stromal lymphopoietin in the development of rheumatoid arthritis. Also, thymic stromal lymphopoietin injection significantly increased the severity of arthritis in mice with collagen-induced arthritis, leading to T cell activation and joint destruction. Mast cells and macrophages in the synovium of individuals with rheumatoid arthritis have been proposed to play a role in thymic stromal lymphopoietin levels in the affected joints. In the collagen-induced arthritis model, which is not commonly associated with T helper 2 cells, the inflammatory phase of arthritis that results in tissue damage is driven by the pro-inflammatory cytokines, including tumor necrosis factor, with thymic stromal lymphopoietin also contributing to this process. Taken together, the findings from various studies indicate that thymic stromal lymphopoietin expression could serve as a potential marker for rheumatoid arthritis. Also, targeting thymic stromal lymphopoietin signaling may offer a promising therapeutic approach for the management of rheumatoid arthritis. Ulcerative colitis is a chronic inflammatory bowel disease that is marked by dysregulated immune responses to gut microbiota, which can contribute to the development and persistence of inflammation in the intestine. While the exact cause of ulcerative colitis is not yet completely understood, it's influenced by genetic and environmental factors. Intestinal epithelial cells have a critical function in maintaining intestinal homeostasis, as they regulate the barrier function and the innate immune responses in the gut and modulate the immune responses within the intestine. Excessive activation of dendritic cells can suppress the production of thymic stromal lymphopoietin by intestinal epithelial cells, leading to uncontrolled production of pro-inflammatory cytokines and contributing to the development of the intestinal disorders such as inflammatory bowel disease. In patients with ulcerative colitis, there is a decrease in thymic stromal lymphopoietin messenger RNA levels, which is negatively correlated with disease severity, indicating here a protective role for thymic stromal lymphopoietin in ulcerative colitis. Low levels of thymic stromal lymphopoietin may contribute to the severity of ulcerative colitis. Thymic stromal lymphopoietin's protective role in ulcerative colitis may be attributed to its ability to promote Treg and T helper 2 cell responses, which can inhibit, on the other side, T helper 1 and T helper 17 cell responses, thus suppressing inflammation in this context. Here is another nice paper about the role of thymic stromal lymphopoietin in cutaneous immune-mediated disease. It's published in Frontiers Immunology 2021. However, I will summarize what came in this paper in another video. Taken together, The functions of thymic, stromal, lymphopoietin in various diseases and cellular processes highlight its potential as post-prognostic indicator of disease severity and as a target for therapeutic interventions. I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell if you are interested in getting any new video, and thank you.